Hello everyone, and welcome back to A&P, and we're back into the skeletal muscle chapter. I do apologize for the previous recording. I didn't realize that my microphone was on mute, but on my Mac, the internal microphone on the Mac picked it up, so you were able to hear some, so that's why I cut it off so abruptly. So I'm going to kind of go back a little bit for where we were, just so that I think this is kind of when I noticed that something was not quite right. So far, as I said before, we're just taking a tour of the muscle cell. So we've looked at the different layers that wrap around it. We had the epimysium that wraps around the entire muscle. We had the perimysium that wraps around individual fascicles or bundles of muscle cells. And remember, the term muscle cell can be used interchangeably with the term muscle fiber. And then we had the endomysium, which actually surrounds the individual muscle cell. And a very important aspect of the endomysium is that that's where those myosatellite cells would be found. And they're basically remnants of the stem cells from which the muscle cells come from, the myoblasts, and those myosatellites are used for repair. Then if we went a little bit further in to the inside of the endomysium, we notice that the first structure that we would encounter is the sarcolemma. The sarcolemma is just a fancy way of saying the plasma membrane of a muscle cell. And if we went into that sarcolemma, past the sarcolemma, we would find what we normally call the cytoplasm, or the gunk on the inside of the cell. It's just that since we're talking about muscle, we're going to call it sarcoplasm. So it's just the cytoplasm of the muscle cell. Very important aspect of both the sarcolemma and the sarcoplasm is that this is where the change in membrane potential begins to initiate the contraction. And remember, the work that a muscle must do is that it's going to have to contract. Another feature that I wanted you to remember is that any cell that's at rest, if we were to try to figure out exactly what the charge was, if we were to record the charge or the voltage reading on the inside of a resting cell, that it would always be at -70 millivolts or somewhere around there. But it's going to be a negative number. So remember, -70 millivolts. This is the measurement. If we put electrodes, something that can measure electrical activity on the inside of the cell when it is at rest. And this is important because it's going to change or differ when we get this cell to work. So when we're at rest, we're at 70 millivolts. But a change in that transmembrane potential, a change in this -70 millivolts, it's going to be what initiates the contraction. And we'll talk about that after we take our tour of the muscle cell. So other structures that we'd find in the muscle cells is that we have T-tubules or transverse tubules. The job of those transverse tubules is to propagate the action potential throughout the cell. So remember that change in membrane potential we talked about, where we have at a cell that's at rest is at -70 millivolts. And let's just put that as rest again. Well, when that change is detected, when that electrical activity of rest of -70 millivolts is disturbed, it's changed, that's what we call an action potential. So changing this, going from a negative number, so going from -70 millivolts to a positive number, and that positive number can be anything that's more positive, -70, so we could go +10, +5, +1. That's more positive than a negative number. That is a change going from resting to now we are going up to an action. It's what we call an action potential. And I'm not going to write that out. I'm just going to draw a line here. So the job of the transverse tubules is that once we've decided that we're no longer going to be at rest, there's been some sort of disruptance in the electrical potential, that we're no longer at rest here, that change has to be propagated or sent throughout the cell. So that's the job of the transverse tubules. When they do that, they send that action potential, and this is a term we're going to use quite frequently between this chapter or this unit and the nervous system, that's going to allow the muscles to contract simultaneously. Because it's able to propagate or send that action potential, it has basically the same properties as the sarcolemma. Big take-home message from these last two slides that we've gone over is that a cell that's at rest is at negative 70 millivolts. Anything that changes that voltage to go from a negative number to a positive number, we consider that an action potential. That action potential is going to dictate that some sort of action happens. And since we're talking muscle contractions, the action that we want to take place is a contraction of that muscle, so the actual work or contracting of the muscle itself. And the T-tubules help to send that signal throughout the muscle cell, and then you also have your sarcolemma and sarcoplasm that are the first one to initiate this change in resting potential. So going into more of the internal framework of the muscle, so keep in mind, once again, we're just on a tour. We're just looking at what different structures are, and then we're saying their function. In much the same way that you'd go to the zoo or museum, and you'd notice the placard on there, and you'd read that. That's basically what we're doing. Our tour guide is just telling us exactly what we're going to be looking at throughout our tour and what its job is. And then later on, we'll get this factory started, and we'll show exactly how all of these pieces work together. So the myofibrils are made up of protein filaments called myofilaments, and there are two different types of myofilaments. You have thin filaments and thick filaments. Thin filaments are made out of the protein called actin. In actin, you can kind of see thin. It's got A-C-T-I-N, thin spelled T-H-I-N, so it's just missing an H in there. So actin myosins are the thin filaments. The thick filaments are the myosin filaments. So thick filaments, myosin, thin filaments, actin. Make sure that you have those memorized because your book toggles back and forth between those terms, and I'm going to do the same thing. That thin filaments, actin, thick filaments, myosin. Those myofibrils, those are going to, in parts of them, make up the striations that we see on muscle fibers. These myofibrils are also important because they are the exact site of the contraction. So far on our tour, we've talked about how there has to be a change in the resting potential to generate an action. That change in resting potential that generates an action, we just kind of call it an action potential has been generated. There are structures that will detect that change, the sarcolemma and the sarcoplasm, and there are structures that will propagate that change all across the muscle cell, the T-tubules, and then you have structures that are actually going to have a physical response to that change, i.e., in the case of muscle cells, contraction, and that would be your thick filaments, myosin, and your thin filaments, actin. Also, what we find here is your sarcoplasmic reticulum. It's kind of like your endoplasmic reticulum. It's just a little bit different. It's a membranous structure that's around all of your thin and your thick filaments or your myofibrils. Just like the transverse tubule, it helps to transmit the action potential or propagate the action potential. The way that the sarcoplasmic reticulum propagates or sends this action potential is that it's going to have a direct contact with the myofibrils. And remember, when we say myofibrils, we're talking actin and myosin. The sarcoplasmic reticulum actually joins up with the transverse tubules, and it forms what we call the terminal cisternae. So you have two ends of the sarcoplasmic reticulum, and then in the middle of them, you're going to have a T-tubule. And what happens is that there is a chemical that is released from that T-tubule that's stored in the sarcoplasmic reticulum that's going to be released onto the myofibrils, the actin and the myosin filaments, that propagates this change. So far, we're just talking about what are the players when we get a change in membrane potential, when we go from transmembrane potential, when we go from a negative 70 millivolts to something more positive. And going from negative 70 millivolts to something more positive is what we call an action potential. So far, we talked about the role of the sarcoplasm, the sarcolemma, the transverse tubules, the sarcoplasmic reticulum, and even the myofibrils of the actin and myosin. All of those players are important for getting this contraction or this overall action to take place in the muscle cell. And we will talk about in depth exactly how that happens. So that triad is really just two terminal cisternaes, or the outpocketing of your sarcoplasmic reticulum, that's what SR stands for, and one T-tubule. Triad, tricycle, three the tri-county area, 3 counties that are kind of closely linked in there together. So triad are 3 parts, 2 terminal cisternae from your sarcoplasmic reticulum and 1 T-tubule. In the cisternae of the sarcoplasmic reticulum, there are calcium. Calcium is vitally important for muscle contractions. In fact, without the calcium that's available, the myosin, thick filaments, and the actin, thin filaments, can't interact with one another. It is the interaction between the thin filaments, actin, and the thick filaments, myosin, that allows for a contraction to take place. So in the terminal cisternae of the sarcoplasmic reticulum, you have concentration of calcium. When calcium is released, the muscle can contract because calcium directly allows the actin and myosin filaments to interact with one another. And the interaction of the actin and myosin, those myofibrils, is what allows for a contraction to take place. So far we've talked about a couple of really important roles of calcium this semester already. We've talked about the role of calcium in bone, that calcium phosphate is responsible for, or hydroxyapatite, for the kind of hardening of the bone itself. We've talked about how bone stores calcium. We also talked about how vitamin D is essential for the absorption of calcium. And now we're talking about the role of calcium in skeletal muscle contractions. So in order for a skeletal muscle to contract, it has to have calcium. Without calcium, no skeletal muscle contraction. So here's a beautiful picture of all the things that we just talked about. So here we have our muscle fiber, which is another name for a muscle cell. We can use those interchangeably. We have all of these mitochondria that are in here. There's like little eggs with zigzags in them. And then the little pink dots, those are those myofibrils. So those are the actin and myosin filaments. So let's look at all this stuff up close. So this blue structure here, this is your sarcoplasmic reticulum. The bulging of the blue part on either side of this yellow thing is the terminal cisternae. The yellow part is your T-tubule, and the combination of 1 terminal cisternae, 2 terminal cisternaes, and 1 T-tubule is your triad. The calcium is held right here in those terminal cisternae and those swellings, if you will, right to the left and right of the yellow structure of the T-tubule. And the T-tubule will release that calcium through this tunnel, through this tube. That's why it's called tubule. If we look at the myofibrils up close, we notice that we have the pink lines, which are your thin filaments, and then the purple lines are your thick filaments. And then in the middle, you have a line that's going to be called your M line. We'll discuss that in a second. Once calcium is released, and notice how the T-tubule is organized to release calcium right where the thin filaments, pink lines, interact with the purple lines, the thick filaments. And because of that release of calcium there, that's going to be what allows for the thin and the thick filaments to interact with one another. A sarcomere is a contractile unit of muscle. That's how we would define that. This would be an excellent fill-in-the-blank question because we're going to talk about sarcomeres a lot this unit. The sarcomere is the structural unit of the myofibrils, and because of the way the myofibrils are arranged, or thin and thick filaments, aka myofibrils, are arranged, they give you patterns on there. And that striped or striated patterns of the myofibrils, those are the striations that we see when we look at muscle tissue underneath a microscope. We look at skeletal muscle tissue under a microscope. So you have these thick filaments, which are going to give you darker bands, and the thick filaments, when we put them in this banding patternation, they're in the A band, and the light filaments are in the I band. So there's a lot of things that we can call these filaments that are responsible for the contraction of the muscle. First, we can call them thin filaments and thick filaments. Then we can call them thin filaments, can also be called actin. Thick filaments can also become myosin. Where we are now is that if we're looking at a contractile unit of muscle, a sarcomere, within that sarcomere we have different zones or bands. In the A band is where you're going to find all of your thick filaments, and they're going to be closest to the center of the sarcomere. In your I band is where you're going to find your thin filaments. A good way to remember this, remember we talked about how actin kind of looked like the word thin, missing the H? Well, actin, thin filaments, they all have an I in them. So they're the I bands there. So that might be something that's helpful to get you to understand these different zones or bands and what myofibril they directly correspond to. In the center of the sarcomere, you have the M line. The M line is going to be in the center of the A band. And what myofibrils are found in the A band? The thick filaments or the thin filaments? Which ones are in the A band? A band, remember we had I in thin, I in actin. We didn't have that for the other word. What was the other one called? We had actin and what's the M word? Myosin, and the myosin are the thick ones. So let's go back here and I'll show you again. So the thick filaments are in the A band, and another term we can use for thick filaments were myosin. So absolutely right, Alyssa. So the A band is where we have those myosins. So in the center of the A band is your M line. The M line is in the middle. So M for middle, M for M line. At the end of the sarcomere, you have the Z lines. The Z lines are going to be in the center of the I bands. So now, which myofibrils, thick filaments, thin filaments, are found in the I bands? Absolutely right. Now we have our thin filaments there. So I'm going to draw a very bad picture of kind of the sarcomere and the points that we've talked about so far. So in the middle, I have my M line. And I'm going to change colors here every now and then. So coming out of that M line, oh dear. Okay, there we go. In our A band, we have our thick filaments. So it's right in the middle, or the center of my A band. So this area here would be my A band. Let's change colors one more time. Let's go with red here. So then out this way, these would be my thin filaments that are in red. And we have, they overlap with the thick filaments just a little bit. So there's a little bit of overlapping that happens there. And then at the end of my thin filaments, these red ones that we're drawing here, and I'll just turn this back black, we have our Z lines. And from Z line to Z line, ooh, that's a horrible Z line, is a sarcomere. So this is a unit of a contractile unit called a sarcomere. Now, all of this stuff that we're talking about on our tour is really important to understand because it helps us to figure out exactly what happens in skeletal muscle contraction. So I'm also going to draw on this side more of my thin filaments because right next door would be another sarcomere. So just as and so from here to here, that's an I band. So in much the same way that the M line is the center of the A band, the Z lines are the center of the I bands. And that what we have going on here is that if I were to draw the rest of this, there would be a sarcomere there, and there would be a sarcomere there, but the only one that we've completely drawn is this sarcomere here. So if I were to put another M line in this area and put another set of thin filaments and some thick filaments, then you'd have another sarcomere. Your book has a much nicer picture of that. We have the zones of overlap. That's where your thin and your thick filaments are going to overlap with one another. The H band is a very narrow area around the M line where it only has thick filaments because the myosin are actually connected directly to that M line. At the ends of the sarcomere, you have these titin fibers, and the job of the titin fibers, as we have highlighted, is to stabilize the filaments. Keep in mind that the sarcomere that we just drew with all of those thin filaments, thick filaments, M lines, Z lines, and all that good stuff, that's going to move because it's going to actually contract. So when it relaxes and goes back to its resting length, we want everything to go back to about the place where it belongs. So you have these titin fibers that help to stabilize the sarcomere when it goes from contraction to relaxation. Now here is a much nicer picture of what I attempted to draw for you all. So notice that from Z-line to Z-line, it's called a sarcomere. Zones of overlap would be where the thin filaments, the pink ones, are going to meet with the purple filaments or the myosin filaments. The M line is right in the center of the A band where all your thick filaments or myosin filaments are found. That H band is just that little narrow area right there in the middle where there are only myosin filaments. You have these green squiggly things. Those are the titin fibers that help to stabilize everything, and they're connected directly to the myosin. And then you have your thin filaments, the little pink lines that are connected directly to the Z lines, and they help for stabilization as well. And in the I band, that's where we only have those thin filaments and the titin fibers. And in the zones of overlap, thin filaments and thick filaments together. For your exam, I won't ask you to label anything like this, but it is important to know. I will ask you questions on which fibers are found in the I band, what's found in the H band, what's found in the A band. So if you can draw it and that helps you to memorize that, then that's perfectly fine. But I won't ask you to label it on your exam, but I'll definitely ask you questions about it. So this is kind of an artist's rendition of a sarcomere, and this is what a sarcomere would look like underneath a microscope, where we still have all of those portions there. I band, only thick filaments. A band, we have our thick filaments in there. H band, only thick filaments. On the middle of that A band, you have your M line, and then you have your Z lines, and from Z line to Z line is a sarcomere, and then you have your zones of overlap where you have both thin and thick filaments together. The zone of overlap is super important because it is the communication or the interaction between the thin and the thick filament, or the actin and the myosin filaments, is what constitutes a contraction. So this picture is really just meant to show you if you were to make a cross-section of the sarcomere at any point in time, what you would find. If you made a cross-section of the sarcomere up the Z line, you would find the titin fibers, the little green ones, and then these pink are your actin filaments. If we were to make a cross-section in the I band, you would be able to see the titin filaments as well as the thin filaments again. At the M line, you would just be able to really see the thick filaments, and if we were over here in the H band, you'd see some of those titin fibers as well. And in the zones of overlap, you kind of see a little bit of everything. You have the thin filaments, pink, the thick filaments, purple, the titin fibers, green. So here's just another picture to look at that, kind of to keep your mind and an understanding of where we are in space and time. So let's start back from the beginning. We have our skeletal muscle itself that is going to be surrounded by, you have your epimysium. That epimysium surrounds these bundles of fascicles. So each one of these bundles are several muscle cells together, and that's your perimysium that surrounds it. So this would be perimysium that's surrounding each of these bundles of muscle cells. So this is a muscle cell, that's a muscle cell, that's a muscle cell, that's a muscle cell, and so forth. If we took one of these muscle cells, aka muscle fibers, out, we see that it's surrounded by the endomysium, and then these red dots in here are the individual myofibrils, or those actin, myosin filaments that we just talked about. And if we were to pull one of those myofibrils out, we'd notice that each myofibril has several sarcomere that are connected to one another, and that each of those sarcomere have actin and myosin filaments in a zone of overlap where they will directly interact with one another. They have myosin of myofibrils in the A band. They'll have actin myofibrils in the I band. They're titin fibers that help stabilize it. From Z line to Z line is one sarcomere, and so forth. So here's that myofibril. From Z line to C line, we call it a sarcomere. Then we have our triad with our one terminal cisternae, two terminal cisternae, and then the T-tubule. And the T-tubule is going to release the calcium that's being held in the Sarcoplasmic Reticulum, in the terminal cisternae of the Sarcoplasmic Reticulum, and it releases it over the zones of overlap. And if we want to get even closer, we'd see those bands again, our A band, our M line, our H band that's right there on either side of that M line, our Z lines that denote Z-line to Z-line is one sarcomere, and then you have your I-band with where your thin filaments are found. So it's just kind of a review of the levels of organization. Remember, we still have not directly talked about exactly how a contraction takes place. So far, we've only looked at the structures that are important for a contraction to take place. And we even talked about the voltage that is there when a cell is at rest. And we talked about what we would call a cell that is contracting or going from that negative number to a positive number, and that's called an action potential. But right now, we're just on a tour, so we're just looking at a lot of these cellular structures and looking at the roles of those cellular structures. So the job of the sarcomere is for it to overlap, or I'm sorry, for it to contract. So the place what allows for that contraction to take place is because you have the zones of overlap where the myosin and the actin filaments can interact with one another. The actin and myosin filaments cannot interact with one another until calcium is released. Well, where is calcium stored? In the terminal cisternae of the Sarcoplasmic Reticulum. Well, how does calcium get out of the terminal cisternae of the Sarcoplasmic Reticulum? It's going to go through the T-tubules. So when that calcium is released, and where is it released again? Near the zones of overlap on your sarcomere. What's a sarcomere again? A contractile unit of muscle. When that calcium is released, then the thick and the thin filaments, aka actin and myosin, can interact with one another. So that's why it's so important to really get the terminology down, and that's why we're spending so much time talking about this terminology, so that when we do get to the muscle contraction portion of these notes, that you have an idea of who we're talking about. And I'm going to, sounds like I'm repeating myself, but I'm going to continue to repeat myself probably throughout the duration of this chapter, because a lot of it is just repetition. The more you hear it, the more you're able to memorize it. And when you get past the first level of learning of just strict memorization, then you can go to the next level of understanding. So muscle contraction, once again, is caused by the interactions of the thin and the thick filaments. What mineral is directly responsible? What has to be released from that triad in order for the thin and the thick filaments to interact with one another? Do you guys remember? It's actually the only mineral we've really talked about today. Same mineral that's responsible in part for making sure that your bone stays nice and hard like we want it to. Absolutely right, Amy, calcium. So calcium has to be released for this interaction to take place. So there are different types of structures that are on the thin filament and the thick filament, so the structures of the protein molecules, they're going to determine how that interaction takes place. So the thin filaments and the thick filaments, they're not just called thin and thick for no reason. They're called thin and thick because of the way that they are structured, what the actual structure is of these proteins. And keep in mind, these suckers are so small, we're talking about proteins here. We're talking about proteins interacting with one another, that you'd have to use a very high-powered microscope, in fact, an electron microscope, to see these guys interacting with one another. So let's start with the thin filament. So the structure of the thin filament is as follows. There are four portions of that thin filament. Even though it's called thin, it's got four parts to it. You should know what all four parts are. First, we have our F-Actin, and that's going to be your globular proteins, or sometimes F-Actin can be called G-Actin, and I call it G-Actin, G-protein. They kind of are interchangeable, so let me just write those down there because I do use those terms interchangeably. So we already have G-Actin, which is the same as G-protein. And what they look like, and I'm going to go back to changing colors, and please forgive me with my mouse drawings. They're not super great, but the best I can do. So you have these twisted rows of globular proteins. So here's one row. And then on the back side of it, you have another row. And they're all kind of touching one another, so don't mind my gaps. All right. Next structure that we're going to look at, so the F-Actin we made red. So F-Actin, G-Actin, G protein, remember all those terms are interchangeable. One thing that you'll find that scientists like to do, they'll find a structure and name it like 4 different things. I think they only do that to confuse people so they can seem really, really smart. But there are multiple names, just like for thin filaments and thick filaments, we could call them actin and myosin. So don't get tripped up on the multiple names that are some of these structures are given. Then we have nebulin, and let's make nebulin green color here. Nebulin is going to hold the G-actin / F-actin / G proteins together. So right here on the butts of our G proteins would be our nebulin strand. So nebulin gets to be green. And then we have tropomyosin. So tropomyosin is a double strand of protein. A double strand is filamentous protein, which means it's like a string. That's what filamentous typically means, like a string a little bit. Let's make that blue. So we'll make this tropomyosin, and it sits on top of the active sites of our G protein. So it kind of goes like this. It just sits right there on the active sites, and then you have kind of a double-stranded sort of protein that's on there. So the tropomyosin strand is blue. And then we are going to make the troponin. Let's make troponin it's like a dark green color. Let's see what color that is. Yes, that'll work. So troponin is going to be right here. Troponin is a globular little protein that's on the active site of the G protein. And it kind of looks like that. And the tropomyosin strand just sits right there on top of them. And the troponin molecule is directly responsible for, it's moved and controlled by calcium. So when calcium binds, and we'll make calcium like an aqua color, when calcium molecules bind with troponin, then it will cause it to roll off of the active site of the G protein, and it exposes it so that the myosin head can directly attach to it. Right now, put that thought on the back burner. Calcium is going to bind with troponin, causes troponin to, tropomyosin actually, to move off of the active site of your G protein, aka F-actin, aka G-actin, and that exposure of the active site is what allows for the interaction between the myosin and the thin filament. For right now, know that you've got the F-actin, the nebulin, the tropomyosin, troponin, know where they are. And there is a much better picture of this that we're going to look at in your notes. And here is that much better picture. So we're not going to pay attention to this so much because I think you guys are pretty good on that. We've talked about it for several minutes now. Now what we're looking at, the red balls, those are your F-actin, G-actin, also called your G protein. The yellow part of those red balls is the active site. Notice that your tropomyosin is that double-stranded filamentous protein. It's green. It's kind of on top of the yellow portion of the G protein, F-actin, G-actin. It's sitting right there on top of it. And then you have troponin, which are those globular proteins that are held in place. If calcium were to be released, that calcium would bind to troponin, cause tropomyosin, the green thing, to roll off of the active site, the yellow part on the red balls, and when that active site is exposed, the myosin can interact with the actin. And when that actin myosin interaction takes place, that's what we have as a contraction. It's actually quite beautiful. It's a fascinating setup. As I said before, muscles, it's one of the contractions, the physiology of muscles, like learning all those muscles is totally not fun. But the physiology of the muscle contraction is quite fascinating. So to initiate the contraction, now if you haven't probably already noticed, we're getting pretty close to getting this muscle contraction started. So we're getting close to starting this process. We have now kind of stepped away from the anatomical structures and the parts of the cell that are needed for a contraction, and we've kind of stepped into the land where we're going to start talking about physiology. How does it work? We already established a couple of slides ago that calcium is important. Calcium is released from this terminal cisternae of the Sarcoplasmic Reticulum through the T-tubule. 2 terminal cisternae, 1 T-tubule consists of a triad. That triad is situated right over the zones of overlap between the actin and myosin filaments. Another name for actin is thin, another name for myosin is thick. When that calcium is released, it's going to bind to one of the 4 parts of that actin filament. We discussed how the actin filament is made out of a G protein, could also be called F-Actin or G-actin. On that G protein, it has an active site. We have nebulin, which stabilizes those 2 rows of globular protein, those 2 rows of G protein. We have tropomyosin, which sits on top of the active sites of each one of those G proteins, and we have troponin. Troponin also sits on top of the active sites of each one of those G proteins. When calcium is released from that triad that we just talked about, it's going to bind to troponin. When it binds to troponin, it's going to cause tropomyosin to roll off of the active site of the G protein and allow it to be exposed so that myosin can connect to that active site. So right now, when a muscle cell is at rest, if we were to check the voltage of that muscle cell, it'd be at negative 70 millivolts. If we have some changes that take place, one of the changes that's going to take place is that the sarcoplasmic reticulum will be triggered to release calcium. When that calcium is released, it binds to troponin, causes tropomyosin to roll over, exposes that active site. When the myosin head sees that active site, it automatically wants to attach to it. And when it attaches to it, we're going to have an action, or we're going to have contraction take place. So we're getting into it. We haven't talked about what happens at the very beginning of a muscle contraction. We haven't talked about what happens at the very end of it. And we really haven't talked about what goes on in the middle, but we're starting to introduce how a contraction takes place. Why is calcium important? Why is troponin important? Why is tropomyosin important? Why is myosin important? Why are the zones of overlap important? So they all have a very unique and important role to play. So the 6 filaments, we looked at the structure of the thin filaments. F-Actin / G-actin / G protein, we had nebulin, we had tropomyosin, and we had troponin. So all those are parts of the thin filament. Those are those structures of it. I kind of give you a little teaser in there about the muscle contractions. I just get so excited talking about it. Yay! Uber nerd moment. And then for our 6 filaments, it's really just twisted myosin subunits. The titin fibers allow for the myosin to kind of connect to the Z-line. The myosin molecule really only has 2 parts. It has a tail, and it binds to other myosin molecules, and it has a head. This head is the part that is attracted to the exposed active site on the thin filament, or the exposed active site on the F-Actin / G-actin / G protein. So that exposed active site is what the myosin head will attach to. And it's going to always reach to its nearest exposed active site on the thin filament. So even though it's thicker than a thin filament, it's deceptively simple. So really not a whole lot going on there. You have the myosin head, you have the hinge of that neck portion of the myosin filament, and that's going to give you that hinging motion. And then you have the myosin head. It is this myosin head that is attracted to the active site after it's been exposed on the thin filament. And we can't get the active site exposed unless calcium binds to troponin. So once calcium binds to troponin, then it will cause tropomyosin to roll off of the active site. And once tropomyosin rolls off of the active site is exposed, and this myosin head that we're pointing to is attracted towards to that exposed active site, and we'll get a contraction here. So myosin action. When we are contracting, the myosin heads interact with the thin filaments, and that interaction between the myosin head and the thin filaments is what we call a cross bridge. From that interaction, the myosin heads are going to pivot, and when they pivot, they produce motion, which is characterized by the shortening of the sarcomere, which is that unit of contractions from Z-line to Z-line. That shortening of the sarcomere is what we consider a contraction. So what our sliding filament theory is that the thin filaments are going to slide towards the M-line. They're going to be pulled in towards the M-line. When that happens, the A zone, or the A band, where the thin filaments are found, or the thick filaments are found in the A band, that's going to stay the same. However, the Z lines are going to move together, that sarcomere, effectively shortening the sarcomere. So the sliding filament theory basically says that the thin filaments are going to be pulled in towards the center, and it causes the sarcomere to shorten. Now, we consider this a theory because, keep in mind, we're talking about proteins, little teeny tiny proteins that are made out of amino acids that are very, very small, that you can't use any of the light microscopes we have here on campus to see any of these little teeny tiny proteins. So we call it a sliding filament theory because this is what we pretty much think is what's happened. But one of you guys might be the one to show us that maybe it doesn't work exactly this way, but we've got pretty good evidence and pretty good biochemical evidence and observations that support this theory. So what we would look for, we have our A band and we have our I band. These Z lines are going to come closer together when the thick filaments pull the thin filaments in towards the M line or the center. So the sarcomere shortens. Resting, contracting. Resting, contracting. So notice how your A band stays the same, but your Z lines are going to come closer to one another. So your I band is going to change length, but your A band stays the same because the myosin fibers aren't moving in towards the M line. The thin filaments, or the actin filaments, or the actin fibers, are moving in towards the thin line. So let's get this thing started. So we've gone on our tour and we have talked about all of the players and what all of their jobs are. Now we're going to start this muscle cell. We're not going to start it from the beginning. We're kind of going to start it in the middle of contraction. The beginning of the contraction is going to revolve neural stimulation. So remember we talked about in the very first lecture that nerves have to send the information that tells it to contract. So raising your hand, scratching yourself, squatting, whatever it is you're doing, your skeletal muscle cells are under your under your voluntary control that you tell them what to do. So we know there has to be some sort of neural stimulation. We haven't talked about specifically what that neural stimulation consists of, but we know that the nerves are going to have to tell the muscle cell to do some stuff to contract. So neural stimulation is the first thing that happens. We'll talk about how that neural stimulation takes place in just a little bit. But right now, since we've already been kind of sticking our toes in the water of how this contraction takes place, we're going to get into the middle portion of it first. So after we have a neural stimulation take place and it generates an action potential, that action potential is going to tell the cisternae of the Sarcoplasmic Reticulum to release calcium. When calcium is released, it's going to bind to the troponin molecule and cause tropomyosin to roll off of the active site of the G protein, aka F-Actin, aka G-actin, and when the active site is exposed, the myosin head will be attracted to that thin filament, that active site on the G protein, and it pulls the thin filament in towards the M line, and that's what constitutes a contraction. ATP is then going to be used at the end of this process to recock the myosin head and for this muscle cell, the muscle fiber, the sarcomere, to be relaxed. So here is kind of a little overview of that muscle contraction. We have a neural stimulation, so the nerves come in and they're going to release a signal that says muscle contract, and that signal that gets released is in the form of a neurotransmitter, and we'll talk about that name of that neurotransmitter a little bit later. That neurotransmitter that's released tells the muscle cell, I need a contraction to take place, and that contraction begins when calcium is released from the sarcoplasmic reticulum. The thin and the thick filaments can interact with one another. Energy is going to be consumed at the end of that process, but when the thick filaments and the thin filaments interact with one another, the sarcomere shortens, and not just one sarcomere on the muscle fiber shortens, but all of them will shorten, and that leads to tension or the contraction itself. So that is where we will end for the day. I do want to show you guys some animations, and we will go over these animations in lab class this afternoon, I want to show you guys these animations real quick, and we'll talk about them in lab. And then I also will talk about them again in our next session we have with one another on Thursday. So I'm going to share with you all my desktop. And let's go to Blackboard. And then here on Blackboard in the lecture lab content folder, we have the lecture notes. And in our lecture notes for muscle notes and animations, we have cross-bridge cycling, excitation, contraction, coupling, which is the one that we've just talked about here, that you should be able to click it and download it, and it will play for you, and you can look at that. You can watch those animations, and they're going to talk about all of the same structures and same things that we've talked about so far. And I'm not going to play the animation here because I haven't been able to figure out how to get the animation to play with the sound and you guys can see it, because I could see it in here, but other you all, the students, couldn't see it on that end. So make sure that you look at these animations, and we will look at them this evening in class. Also, what we have is I have the steps, because you can probably best believe that the steps involved in skeletal muscle contraction, that's going to be a short answer question on your next exam. So all of those steps, your book separates them down into like 5 steps or something. I have a different way that I've separated them down. Remember, I don't care how many steps you use, just as long as you talk about all of these portions of it. We haven't gone over everything yet. We've really only talked about from step 4 to the top part of step 5. So that's really all we've gotten to. But we will talk about steps 1 through 3 and steps 6 and 7 again. So we just started kind of in the middle because we were already talking about actin, myosin filaments, and so forth. All right, questions, comments, or concerns? Are you all as fired up about skeletal muscle contraction as I am? I know you are. I know you're super excited. In lab today for my Monday class for you all, we're just going to be continuing with what we left off with last week. So last week we did the bones and muscles of the lower extremities. We're going to look at those bones and muscles of the upper extremities. So your goal in lab will be to find those bones and muscles. The joint that you will look at is a shoulder joint. The shoulder joint in your book is not very good, so make sure you look at the pictures on Blackboard. Use this as a reference because I have an actual shoulder joint there that's labeled. And then also review the things that we looked at, the bones and muscles of the lower extremities as well. All right, so we don't have any questions, comments, or concerns. I will see you all later. Enjoy the remainder of your morning.